Hello and welcome to an introduction to hydro-ecological modelling using the HE Toolkit package. This is the second of two workshops which provide training in the use of the Environment Agency's new Hydro-Ecology Toolkit, which is an open source R package for assembling, processing and analysing hydro-ecological data. My name's Andy Davey. I'm a Principal Data Scientist in the Hydro-Ecology team at APEM. I'm a freshwater ecologist by training, and I've experience in water resources, water quality and catchment management in various research and consultancy roles. Since 2021, I've been working very closely with the Environment Agency to develop the HE Toolkit, working alongside my co-presenter today, Mike Dunbar, who is the former team leader of the Environment Agency's National Hydro-Ecology team. And it was Mike's pioneering research into hydro-ecological modelling which provided the impetus for developing the HE Toolkit. And it was Mike who was responsible for commissioning and designing the toolkit. As I mentioned, this is the second of two workshops. The first workshop provided a general introduction to the HE Toolkit and was aimed at users who wish to work with open source environmental data. It covered various techniques for importing, cleaning, visualizing that data to provide an automated workstream. That workshop took things as far as data visualization. The second workshop really picks up and builds on workshop one and is aimed at more advanced users who want to use statistical techniques to build predictive models that we can use to help assess ecological responses to changes in flow. This second workshop, we're going to begin with a little bit of an initial background, just talk a little bit about what hydro-ecological modelling is. I'll then provide a little bit of theory about some of the statistical methods that we'll be using. So this is very much a crash course to introduce you to linear mixed effects models. We'll then recap on some of the material that we covered in workshop one on data assembly, looking particularly at some sort of tips and advice for processing hydrological and ecological data sets. We'll then look at how we can bring all of that data together and format it in a way that makes it suitable for hydro-ecological modelling. We'll then look at some different approaches for model fitting, model selection and model diagnostics, with the goal of developing robust models that we can use for prediction. Finally, we'll wrap up by talking briefly about how these models can be used in a real-world setting to help us make water resources decisions. This workshop is accompanied by a series of practical exercises, which we've made available as an RStudio project and hosted in a web-based format on POSIT Cloud. A link to taking you to that POSIT Cloud repository accompanies this video. The remainder of this workshop is split into a series of tutorials where we look at how we can use different functions from the HE Toolkit to import, process and analyse hydro-ecological data. However, to do more than just describe how the HE Toolkit works, we've also set up a series of practical exercises which give you, the user, an opportunity to get some hands-on experience of using the HE Toolkit and also some example code that you can use as a template for developing your own workflows. To access these practicals, we've developed or set up an RStudio project which is hosted in the cloud, which you can access via the link which accompanies this video. To give you a quick tour of that online project, I'm just going to give you a quick walkthrough now. So accessing the link, it takes you through to the POSIT Cloud page. POSIT Cloud is just the new name for RStudio, and this is a cloud-based version of the familiar RStudio environment. We'll need to register for a free POSIT Cloud account. Logging into your account will then take you through to the practical workshop. You'll notice that will just take a few minutes for all of that to upload. And when it does, you'll see basically on screen in your web browser the familiar RStudio development environment. To accompany each workshop, we've created a set of example data sets and also some example R scripts, which illustrate all of the different functions that we're covering in these workshops. Here on screen now, you can see we've got the console over here on the left. We've got the environment listing the various objects that we create on the right. We've got a files list as well down here in the bottom right-hand corner. Once you've successfully logged in to access the project, the first thing to do is just to save a permanent copy. It simply clones a copy of the RStudio project over to your own personal workspace, which means you can work on it, change it, and save your edits. Again, that will just take a few seconds to clone across. There we go. And now we can just start to explore some of the files which we've made available for you here. The data folder contains all of the example data sets that we have created for use in these practicals. So if you want to, you can zoom into there. This particular example, we've got a CSV file containing some flow data. We've got some previously saved invertebrate data, and we've also got a metadata file, which lists out the biology and flow sites that we're going to analyze. We've got an R script as well, which provides some example code you can use if you were to install the HE Toolkit from scratch in your own new RStudio project. For the purpose of these practical exercises, however, the HE Toolkit and all its dependent packages and any other packages that we've used in these practical exercises have already been installed in the project here. So there's no need for you to download those from scratch. Instead, you can just navigate directly to one of the R scripts which contains the code for each of the practical exercises. So for example, we can click on practical exercise 1 and that loads the script for us in the top left-hand corner here. All we need to do to start analysing our data is to load the HE Toolkit using the library command. So I can click onto row 6 and use the run button there. Then we can step through the script a line at a time or a section at a time and run the code. Outputs will then be returned in the console in the bottom left, and any plots will appear under the Plots tab on the right. Scripts are structured into a series of sections. So this first practical exercise, for example, first of all allows us to load the HE Toolkit, read in some metadata, and do some initial setting up to provide a list of all of the site IDs. The second part of the script then imports some flow data. The next part of the script imports the environmental data. The next section imports macroinvertebrate data. We then import RHS data. Together, all of these sections can be linked together, so you can run the script either a line at a time, or if you want to, you can select multiple lines and run those together. You will see here, for example, that some of the tables of data that the script uses will pop up at the top here, so you can inspect those. It looks rather like an Excel spreadsheet. And any objects that you create, any data frames or model objects, will be listed out here in your environment. If you want to inspect an object that you created, a data table, for example, then you can click on the table icon on the right here, and again, that will bring up a copy of that data table for you to inspect visually. Feel free to open up, run, and if you're feeling ambitious, please feel free to sort of tinker with and edit any of those scripts. It gives you an opportunity to see a typical workflow, and hopefully this will provide you with a useful template for doing your own analyses. Before we launch into the details of different statistical modelling techniques, I'd like to begin with a quick introduction to explain what hydroecological modelling is and look at how we can use the HE Toolkit to help build such models. So what is hydroecological modelling? Here I've defined it as being the calibration of statistical relationships between hydrological and ecological metrics, and then applying those relationships to help us understand and predict ecological responses to changes in flow. Basically, we can use any ecological metric we like. This could be, for example, a macroinvertebrate life score, a measure of fish density, macrophyte cover, or some measure of algal abundance. So long as it's a univariate variable, that is, the numbers can be represented by a single column of data, then we can apply regression modelling techniques to explore how those metrics respond to the flow conditions that those organisms have experienced during their lifetimes. You can, of course, also analyse multivariate data if you have, for example, abundances of different species within a community, but that relies on different multivariate statistical techniques, which we're not going to cover in today's workshop. As you can see from the two diagrams on the slide here, there's at least two different ways in which we can explore these hydroecological relationships. On the left, there's an illustration really of how you can correlate your ecological metrical response with natural hydrological variation. So essentially here we're comparing our metric scores under wetter and drier conditions. So we're looking at how the ecology changes over time in response to changes in flow at each site. Alternatively, as shown on the right, we can directly assess the effect of flow alteration by comparing sites which have more or less natural flow regimes. Here the focus is on comparing our metric scores across different sites, so this is more of a spatial comparison. Of course, it is possible to have a combination of those approaches where we seek to model both temporal patterns and spatial patterns in our data. Analysing spatial patterns in our data and to assess the effect of hydrological alteration does rely on the use of hydrological modelling tools to characterise the natural flow conditions at each location. That provides a reference point against which we can assess the historical flow conditions and see how much the flow regime has been altered by abstractions, discharges and impoundments. That obviously requires some additional hydrological data. So today, to keep things simple, we're going to focus on modelling the effects of hydrological history because that relies solely on using readily available gauged flow data. Obviously, hydroecological models focus particularly on understanding the effects of flow, but being statistical regression models, there's nothing to stop us adding in other explanatory or predictor variables into our model. This can be useful, for example, if we want to be able to control for other confounding pressures which might be acting upon our ecological community. So, for example, we might choose to incorporate information that characterises the in-stream or riparian habitat conditions at each location, or information about the wider catchment characteristics, or possibly we could also integrate water quality information. So, although today we're going to be focusing very much on understanding the effects of flow, these models can be elaborated and made more sophisticated to start to disentangle the effects of those different pressures. Now, hydroecological models are useful because they provide a data-driven approach to understanding the effects of changing flow regimes. There's nothing to stop us taking hydrological and ecological data and building, for example, a HEV plot, as we looked at in workshop 1, and that gives us a quick visual assessment of the data. We can also, of course, rely on expert judgment and our experiences of analysing data from other similar rivers, for example, to help us judge what the impact might be of a particular change in flow. Hydroecological models, though, because they're data-driven, they allow us to make much more quantitative predictions. We can indicate not only whether a change in flow might have a positive or negative impact, but we can make a quantitative prediction exactly how much change we would expect to see. Hydroecological models also provide a mechanism for testing specific hypotheses and for trying to understand which aspects of the flow regime are most important in driving patterns that we see in our ecological data. When we develop a model, we calibrate it based upon data from one or more sampling locations. So by its very nature, these models are tailored to the specific local context that we take our data from. Hydroecological models are great, but the process of assembling all that data that we need, processing into a suitable format, doing some exploratory visualization and then modelling it, that can be a relatively slow and time-consuming process, and it does require a certain amount of technical capability. The other problem we face is often we might be interested in trying to assess the effects of flow changes at one particular location. And if we're lucky, that location will have a lot of ecological sample data spanning several years, ideally decades. However, that's often not the case. And so we may be faced with a situation where we have a relative paucity or maybe even no ecological data at all for the location that we're interested in assessing. So instead, what we can do is take data from multiple different sampling locations, so long as they come from rivers or watercourses which have fundamentally similar hydrological and ecological characteristics. And we can build a multi-site model. This has the benefit of giving us a much larger volume of data on which to calibrate a model, and allows us to make inferences as well and provide essentially better predictions for locations which have limited monitoring data, and also allows us to extrapolate and make predictions for locations which have no ecological sample data at all. And these models, once calibrated, can be used then to predict exactly how the ecological community, as represented by our response variable, will change under different abstraction or flow scenarios. So the HE toolkit really is just to help, speed up, automate, standardise that whole process. And it's aimed really at hydroecologists who might be working in water companies, within regulatory organisations, maybe in consultants or the academic sector. Basically anyone who's interested in working with these kinds of open source environmental data sets and wants to build predictive models. As I mentioned earlier, this modelling approach can be applied to any biological receptor of interest. However, research to date and real-world applications have tended to focus predominantly on benthic macroinvertebrates, partly because they are very widely monitored, also because they have well-established community-level metrics such as life and WHPT, and also because those metrics, due to the way they're designed, do respond quite nicely to antecedent flow conditions. So the examples we're going to be looking at today will focus on macroinvertebrates, but please just bear in mind that these approaches can be applied to any kind of biological data. This flowchart will look familiar to you if you've already covered workshop 1. It maps out as a workflow all the different functions within the HE toolkit, those are the green boxes, and shows how they link together. So we're basically on a journey here from various different input data sets, shown in blue on the left-hand side. And in workshop 1, we looked at a variety of functions which allow us to automatically import data from different open source data sets and online databases. We then looked at various different data processing techniques for visualizing, cleaning, summarizing and joining that data. And we also looked at HEV plots as a way of visualizing time series of hydroecological data. What we're going to look at now in workshop 2 is we're going to start off with a brief recap of some data processing functions that we covered in workshop 1, but look at a little bit more detail and think a little bit more about how we can tailor those to work with different kinds of data. We're going to then look at a couple more data exploration functions which allow us to assess how suitable our data set is for hydroecological modelling, and then look at some functions which can help us to help the modelling process, essentially, to help decide which models are the best, and also to determine how robust our models are meeting the different statistical assumptions.